Hello. So I'm going to show you how to do this lab where you're going to watch transcription and translation happen. And you're going to figure out what is the difference between the different tubes that you're going to set up. So you're going to have 4 tubes, and they're connected together in this little strip. And at the base of these tubes is a little pellet called a biodot. And that's a dried pellet of all the enzymes needed to do these reactions, and ribosomes and stuff. So it's called it's like a artificial cell. There's no cell, it just has all the stuff that cells have in them. And then you have 4 separate tubes that have your reagents in them. Okay, so for your strip, they're all going to have biodots in them. So here's your strip. You're going to have 1, 2, 3, 4 tubes. 1, 2, 3, 4 in this strip. They're all going to have biodots. And I want to make sure that sometimes the biodots get kind of knocked up to the top of the tube. So you want to make sure that you kind of knock those down so they're at the bottom of the tube. So just kind of notice that. So the next thing in there, for tube 1, you're just going to have water, and you're going to put 8 microliters of water. And this water is special water because it doesn't have any nucleases in it, because we don't want, sometimes on your skin or even in the air, there can be some enzymes that break down DNA and RNA, so we want to use the special water that I've given you. So 8 microliters of water in tube 1, that's your control. In tube 2, you're going to put 4 microliters of the water and 4 microliters of DNA A, so the type A DNA. For tube 3, you're going to put 4 microliters of water and 4 microliters of DNA B. And in tube 4, you're going to put 4 microliters of water, 4 microliters of DNA A, and 4 microliters of kanamycin. So the kanamycin is an antibiotic that inhibits ribosomes. And so this is going to prevent translation. Okay? So what you can do first is put the water in all the tubes, because then you don't have to change tips. As long as you don't touch, well, you try not to touch the tip to the pellet, to the biodot. So you can put 8 microliters of water in the first, and then 4 microliters of water in all of the other 3 tubes. So that's what I'm going to do right now. Let me show you my setup. So here I have my tip box, but you can see I've also used it to hold all my tubes. So I have my strip, and then I have my 4 reagents: DNA A, DNA B, kanamycin, and water. You may be given a P10 micropipetter, or you may be given a P20. So you want to look on your micropipetter to see what the range is. It's usually written on the side or on the top. And so like mine right here says 2 to 20. That means this is a P20. It goes up to 20, but you can't measure anything less than 2. This one's a P10. So you can see it says 10 here. So this will measure anything between 1 and 10 microliters. If you're using a P10, you have these tips, these smaller tips that I'm going to give you. They're usually white. And if you're using a P20, I'll be giving you yellow tips. And they're much bigger. You can see the difference in size of the 2 tips. Okay. So I'm going to go ahead and use my P10 because I love P10s. They're just really nice and accurate. But I don't have 16 P10s, so not all of you are going to get P10s. Okay. Now my P10 has is very accurate, so it goes down to the hundredths place. But you might have one that only goes to the tenths place. So I'm going to set my P10 right now to 8 microliters because I'm going to add the water. So I'm going to show you what a P10 looks like with 8 microliters. So this one is 8.00. There's a line there showing you the decimal. For a P20, 8 microliters, there's going to be one digit that's a different color, and that's your tenths place. So I'm going to change this to 8.0. That's 8.0. So that you can see that the 0 is red. So that's the decimal, that's the tenths place. So that's 8 microliters. Okay, so if you remember, when you're using a micropipetter, there's two places where you can push your thumb. There's the measurement spot, which is the first resistance point, and then there's the sort of push out all the extra liquid stop. So you always want to push to the measurement stop, which is the first place of resistance. So you push down, that's like squeezing an eyedropper, and then I'm going to find my water. So water has a W on it. And it also, just in case that W wears off, it has a dot on the very top of the tube. But be careful not to rub the W off, because then you're going to end up with tubes you don't know what the hell they have in them. So I'm going to push again to that first stop. I'm going to put the tip in, but notice how I'm looking so I can see where the tip is. I want to make sure the tip is in the liquid, and I let go of my thumb to draw it up. So this is, I don't know if you can see that. That's 8 microliters. Okay, so I'm going to cap the tube, put it back, and take my first tube in the strip and put the 8 microliters in. And then I'm going to change this to 4. And I'm going to put 4 microliters of water in all of the other tubes. So push to the first stop, stick it in the liquid, let go, put 1, 2, 3. Okay. And you just kind of want to tap these down, make sure all the liquid's at the bottom. Okay, so I've added the water. So then I use a cup to eject my tips in so that they're not all over the place. All right, so the water's done, check. So now I'm going to put DNA A into both tube 2 and tube 4. So I'm going to have it set on 4 microliters. I'm going to get my DNA A, which is marked with an A. Okay, so there's an A on it, but it also has a mark on the hinge, just in case your A mark wears off. Okay, so it's very important that I measure this carefully because I didn't give you guys enough to measure incorrectly. So I got 4 microliters. I'm going to put that in tube 2. And since I'm going to use A again, and I haven't put anything except water in any of these, I can use the same tip and put another 4 microliters in tube 4. And you want to make sure that you're not flipping this around so you won't know which is 1 and which is 4. And if you're worried about it, you can mark them with a Sharpie, 1, 2, 3, 4. Okay, so I want to throw away my tip, and now I'm going to put DNA B in. I'm just going to put 4 microliters of DNA B. Done with A, I'm done with the water. DNA B obviously has a B on it, and it doesn't have any mark on the lid. So DNA B only goes in tube 3. Okay, so I mark that, put that in tube 3. And then I'm going to put the kanamycin in tube 4, 4 microliters again. Kanamycin has a K, and it has a little mark towards the end of the cap that's opposite of the hinge. So I'll show you where that is. It's marked there. Okay, and that goes in tube 4. And if you forget, just look at, you should write down what goes in which tube so that you don't forget. Okay, then you want to like tap down all the liquids, make sure all the liquids are tapped down. You put the lid strip on the top, and then you're going to put it in your little viewer. Now what I've changed for you is the filter is orange now, so it can see both green and red. Now if you're in a hurry, you can hold on to these tubes like this, and the reaction will happen quicker. But what I did was I just like sat down with my favorite TV show, and I just started a timer, and then I just kept looking at it and looking for any difference that I saw. And when I saw a difference, I wrote down what time it was, and then I took a picture of what I saw, and then I wrote down what I saw like in words. And so it took about, I don't know, about a half hour for me to see the first real change, and then I think as it went on, it was like an hour or so before I saw the second change. And then I kind of went back, and when I was getting ready for bed, and I checked again, and I saw another change, and then I checked again on the next day, and there was even more of a change. So just make sure you're writing down all your observations and the times that you saw them, and take pictures. Okay.